high molecular weight, converted sulfate-glucoglycan, and B97 melanotransferrin are target antigens, potential target antigens, and these molecules are highly expressed on the vast majority of malignant melanomas, and their expression is significant on the human major melanocyte. Single-chain variable fragment is the most important part of the CAR structure. In our case, high molecular weight and B97 specific single-chain variable fragment was constructed by unicorn column, and that their specificity was confirmed by flow cytometry. And in our preliminary data, we found that both are specific for their antigens and their expression were similar. Before discussing the result, I would like to briefly describe how did I generate specific CAR T cell. This process involved in two before steps. First, the process involved in using retrovirus production. There are three different plasmids: CAR, backbone, RD114, and holoplasmid were combined into the viral vector on H293 T cell. Then, following 48 hours incubation, the producer cell line release viral vector encoding CAR that can be collected from the cell culture and used for the next step. During the following process, which is called T-cell transduction, these cells were incubated with the viral vector encoding CAR. The viral vector uses viral machinery attached to the T-cell, enter into the cell, and introduce genetic material in form of RNA, and the RNA, this genetic material is encoded CAR, and the RNA is reverse transcribed into the DNA and permanently integrates into the T-cell genome. Therefore, CAR expression is remain as the cell dividing. Then finally, specific CAR is transcribed and translated by T-cell and expressed on the cell surface. Incorporating the previous described single-chain variable fragment, we clone the first generation CAR that's specific for B97 and high molecular weight melanoma-associated antigens. And single specific CAR T cells were generated by retrovirus production under genetic encoded like both CAR has CH2, CH3 in the hinge region. Therefore, we confirm the transduction efficiency by using LSAT 647 anti-human IgG standing microcytometry. After transduction, 30 to 40% of the T-cell expressed melanoma-specific CARs, and it is visible that B97 and high molecular weight specific CAR expression were similar on the T-cell membrane. Thus, these are the specific CAR T-cells are the candidate for us to go for further experiment to investigate their activation and their cytolytic function. By using confocal microscopy, my supervisor investigated target-dependent synapse formation between miller and specific CAR T-cell, and CARs are also stained with LSA647 conjugated anti-human IgG. And in these microscopic images, we found that anti-B97 specific CAR from significantly larger and more closely synapses than high molecular weight. Thus, we were curious that the difference in the synapse formation has any effect on the T-cell activation. Then the early T-cell activation was investigated by short-term assays co-culture using specific CAR T-cell and miller following 5, 15, 30, and 60-minute incubation with the specific standing of the phosphorylated CD3 zeta LCK ZAP signaling molecule, and by flow cytometry. And this represents the percentage of the phosphorylation of the CAR-positive T-cell following the incubation time. And it is clearly shown that B97 specific CAR T-cell induce faster phosphorylation of CD3 zeta within 5 and 15 minutes than high molecular weight within 30 minutes. Coherent with this B97 specific CAR also induce phosphorylation. Earlier in the B97 specific CAR T cell. For investigating the long-term T cell activation, we perform the interferon gamma releasing assays, and this by using non-transduced T cell anti-ham molecular weight B97 specific CAR as the effectors and the miller as the targets. And we found that anti-B97 specific CAR is secreted the highest level of the interferon gamma. There is no cytokine production in the action of the antigens and in the co-culture with the non-transduced T cell. In summary, both B97 and specific CAR T cell recognize miller target cell, even though B97 and high molecular weight antigens have the similar expression level on the cell surface, and anti-B97 and anti-high molecular weight specific CARs have the same the similar expression on the T cell. Our experimental results strongly highlight that anti-B97 specific CAR induce better, faster T cell activation and from larger synapses. Thank you for your attention.